So hello, my name is Michal Ozery-Flato and I'm a research scientist in the AI for Healthcare department in IBM Research Haifa. And I will talk on identifying drug repurposing candidates from real-world data, focusing on the Parkinson disease case study. So what is drug repurposing? Drug repurposing is the search for new uses for existing drugs. It is motivated by the fact that clinical trials, which are used for approving the drugs, are lengthy, costly, and most often end in failure. So drug repurposing mitigates the risk for failing clinical trials due to safety issues because these drugs were already found to be sufficiently safe. So there are examples of repurposed drugs. You're probably all aware of the story of Viagra, which was originally designed for treating hypertension, but then was shown to have beneficial adverse effects on erectile dysfunction. Another known story is of thalidomide, which was prescribed for pregnant women for easing their morning sickness. However, it resulted in over 10,000 cases of children born with severe deformities. So 40 years after this tragedy, the drug was approved for or repurposed for treating multiple myeloma, which is a type of blood cancer. So there are successful discoveries in drug repurposing, but many of them were due to good luck or following specific pharmacological insight. So clearly there is a need for a systematic approach for drug repurposing. So let us focus on the third phase of clinical trials, which are usually designed as randomized controlled trials. So under this design, each participant is randomly assigned to either the treatment cohort where the tested drug is given, or to a control cohort where an alternative drug or a placebo is given. So randomized controlled trials are the gold standard for evaluating the safety and efficacy of drugs. And the randomization process is the one that ensures that the treatment and control cohorts are statistically similar. So the use of randomized controlled trials is limited for several reasons. They are costly, they are lengthy, and of course there are ethical issues involved. Also, they are considered to be non-real-world settings, and in many cases they are relatively small, so there is insufficient statistical power to detect effects which could be either harmful or beneficial. Also, in many cases, or even in most cases, the follow-up period is relatively short, which results in inability to detect long-term effects. So on the other hand, we have abundant real-world observational data accumulated in electronic health records and claims data, and these track prescribed medication and health outcomes for millions of patients under real-world setting. So in IBM, we have two large datasets of this type. We have the Exploris dataset that covers over 60 million patients, and MarkingScan that covers over 100 million patients. However, inferring the effect of treatment from real-world observational data is challenging due to several reasons. First, treatment assignment is not random, so therefore treatment cohorts are expected to be biased. Another thing is this is a retrospective analysis, so we have no control over treatment duration. Also, there are biases related to loss to follow-up and noise, and also the clinical outcomes that are desired for our studies may not be directly observed. So we need to integrate domain knowledge and also be some creativity in order to devise some proxies for these outcomes. So in IBM, we developed a framework for drug repurposing that addresses these challenges by incorporating domain knowledge with cause and inference methodology that account for the different biases in the data. Our framework for drug repurposing is based on the idea of emulating randomized controlled trials using large databases of observational healthcare data such as Exploris or MarkingScan, but of course other databases can be used as well. So it incorporates domain knowledge via an interface that allows the user to define study design parameters such as the eligibility criteria and the follow-up period, as well as the outcomes that we would like to measure. It also allows the user to define all the potential confounders, which are usually concomitant drugs, comorbidities, and any factor that allows us to reflect the clinical state of the patients. Then the framework automatically scans the database, searching for all the drugs that can be tested, and for each it creates a trial. For this trial, it has tracked the treatment and the control cohorts, as well as all the variables related to the outcomes and confounders. The control cohorts are based on related drugs, drugs for a similar indication that are detected based on the ATC classification system. Then the data of the trial is processed by the drug repurposing engine that emulates randomized control trials by applying a causal inference methodology that account for all the detected biases. And the module that implement the causal inference methodology is an open source library that can be accessed in this URL. Finally, all the trials are run in parallel, and the framework aggregates the different results into a single report. And when we compared results of independent trials that were run on different databases, meaning Explorers and Markers, and we saw a remarkable agreement between the estimated effects, which confirmed or supported the validity of the results. We use this framework for identifying drug repurposing candidates for Parkinson disease in a joint study with Teva Pharmaceutical Industries, and the results of this study were recently published in the journal Frontiers in Pharmacology. So in our study design, we included patients that were diagnosed with Parkinson disease and were over the age of 55, and also applying some other exclusion criteria. Overall, we had around 1,000 patients in each of the databases that were included in our study. The follow-up period was defined as 2 years, and our clinical endpoint was Parkinson dementia onset. So we identified 2 drugs that have a potential disease-modifying effect for Parkinson disease, one of which is rasagiline. This is a drug that is currently indicated for treating PD motor system, meaning that this is a symptomatic drug and not a drug intended for healing or slowing the disease. So there is a mechanistic support for neuroprotective effect of rasagiline, and therefore there have been several studies that try to evaluate this effect, however, without any conclusive results. We think that one of the reasons for this is that the number of participants was not large enough compared to our study, in which the number of participants were 10 times larger, more than 10 times larger than the largest study to date. Also, our follow-up period was longer than the follow-up period of the previous studies. And as you can see here in the Kaplan-Meier plots, the difference between the treatment and the control cohorts increases with time. The second drug that we identified was zolpidem. This is a psycholeptic indicated for sleeping disorders. There are some mechanistic evidence that it can provide some neuroprotective effect. However, all the studies to date, and there were relatively few, were quite small, again, compared to our study, which, as you see, is relatively large. So in conclusion, our study indicated that emulated clinical trial with big observational medical data can identify promising repurposing drug candidates, which merit further clinical analysis and experimental validation. The entire approach is concordant with the vision of the 21st Century Cures Act. This is a US law designed to help to accelerate medical product development and bring new innovations and advances to patients who need them faster and more efficiently. So in future challenge for us is to make the computation more precise, targeting a subpopulation with maximized beneficial effect for the repurposed drugs. And we are very interested in collaborating on drug repurposing and causal inference in general for healthcare. So if you're interested, please approach us. Thank you.